Okay, now we're going to go through the Gram stain lab. All right, you'll be watching a video on this. But Gram stains are the most popular stain we use in the lab. It is a differential stain, meaning that we use more than one color to differentiate something. And the one thing with a Gram stain or any of these differential stains that we do in micro, these things need to be on opposite ends of the color wheel. It doesn't help you if you lay down a violet or a blue stain and then counter stain with a blue. You can't see the difference. So all your differential stains are going to come in with colors that are on opposite sides of the color wheel or something that's like that. So we're going to be using Gram stain, which is using violet here, and we'll be color staining with something over in this range. Okay, so you can easily see it. Gram stain, again, one of the most important stain processes in micro. It can be done in a stat basis. So when somebody comes in and you think they might have, bacterial meningitis, a stat Gram stain will be ordered on that spinal fluid, and we can say, yes, there is bacteria in the spinal fluid, or no, there's not bacteria. And then it's maybe a viral meningitis, okay? So it is used to differentiate these bacteria into two groups, Gram positive or Gram negative. And again, it's based on the cell wall and how it's constructed. We've gone through this in the lecture, but I want to really, really hone in, and I think you'll be able to see this better. So in the lecture, you found out that the difference with the Gram positives and the Gram negatives is the Gram positive have this thick layer of peptidoglycan, very thick, okay? Versus the Gram negatives, which have a thin layer of peptidoglycan, okay? And then on the outside of that, they have that LPS, that lipopolysaccharide that's sitting on the outside, okay? So this can easily be removed, just FYI, okay? So let's look at the Gram stain. So first of all, we make the smear, and you got a video on how to make smears, but basically we take that and we either mix it with water, or if it's liquid, we can just put it straight on the slide, all right? And we lay it down, and you don't see anything except, maybe a wet circle or a little bit of the bacteria smeared on that slide. Okay. So the first thing you do is you apply what's called the primary stain, the first stain. And that primary stain in the Gram stain is crystal violet. It is a basic dye. It stains all the cells purple. Again, it's positively charged, is going to be attracted to the slightly negatively charged bacterial cell walls, so they will sop up this crystal violet, okay? And if you let it stand for a minute and you rinse it with water, if you were to look at the slide at that time, everything's going to be this nice purple color, okay? All right, step 3 is you take that slide and you add the mordant. Think of it as, when you put in bricks, you're laying bricks, you put in mortar. It's the same thing. We're going to flood that smear with iodine, and that iodine forms an insoluble complex with the crystal violet and anchors it to the cell wall. And you let it stand for the same amount of time that you put the crystal violet on. You don't have to do crystal violet at a minute. You can do it for 45 seconds. But whatever time that you're doing the crystal violet for, you got to do the iodine for. Okay, so we said 1 minute, so we're going to allow the iodine to sit a minute. Okay, so everything is still at this point. If I wash this off and look at it, at this rinse, everything's still going to be purple. Because we haven't done anything except to lock in that stain. All right, now is where we start to differentiate and distinguish these organisms. We can use a decolorizer. And the decolorizer is ethanol and acetone. And just 2 or 3 seconds, it doesn't take much. And what it does is remove that outer membrane, that LPS, off those gram-negative bacteria and leaches that complex, that dye complex, that Dye-Morton complex out of the gram-negative cells. So you remove that LPS, and what you have left is your peptidoglycan layer with no dye on it. Okay. Now I will tell you, if you do this too long, I can turn any gram-negative into a any gram-positive into a gram-negative. I mean, you just can. It just leaches. All right. If you leave this on, so you can actually, if you leave the alcohol on too long, methanol on too long, you can leach through this peptidoglycan layer on the gram-positives. But if you've done gram stains long enough, you don't do that. Not to, you just do it for a short period of time. Okay. Then you rinse it again. Again, if we were to look at it underneath the microscope at this time, our gram positives are going to be stained, and our gram negatives, you're not going to be able to see. Okay? Because we've removed that first stain. Then we come in and counter stain. And we're going to flood it with that color that is opposite on the color wheel, the safranin, which is a nice, bright, pink. And let it stand 1 minute again. And what will happen is the peptidoglycan layer of the gram negatives will pick up that safranin. But the gram positives are already stained a dark color, and they're not going to, even if it picks up a little bit of safranin, there's a whole bunch of crystal violet sitting there, and it really can't get in. Okay? So what you get is, then you rinse that off, you blot it dry, and you look at the slide under oil immersion. And what you get is your gram positives are purple and your gram negatives are red. And so when you report out a gram stain, you report out the gram reaction first, and then the shape. So gram positive cocci, and then in chains or in clusters, diplococci, gram negative bacilli. Okay, you can have gram positive bacilli, I assure you. Okay, so your gram positives are going to have a lot of information about the shape that's important, because it'll tell the doctor, do I have a bacillus, which is gram positive bacilli? Do I have a streptococcus, which is gram positive cocci in chains? Or do I have a staph or something like that, which is gram positive cocci in clusters? Or do I have them only in diplococci, which I may have a diplococcus? Okay, so a lot of information. Gram negatives don't report out the shape because they're bacilli. Okay, so there we go. That's it. That's the gram stain.